Today topic is senescence in the plants. What is the senescence? Senescence is age-dependent, progressive and deterioration process at cellular, tissue, organ or organism level. It ultimately leads the death of the organism. It is energy-dependent, genetically programmed aging process. It occurs due to stress-inducing as well as age-related processes. It is essential for the plant growth and the development. Then, pattern of senescence. There are different patterns of senescence. First is the cell senescence, second is tissue senescence, third organ senescence. In organ senescence, there is another three types: shoot senescence, leaf senescence, flower senescence, and fourth is the whole plant senescence. First one is the cell senescence. It shows continuous senescence. Example is scleroid formation in plant. Second है tissue senescence. It is important for tissue differentiation. Example during the cork formation, scleromatous formation, xylogenesis, etc. Organ senescence. It mostly occurs in the polycarpic species, which flower and fruit repeatedly senescence of organ take place at different interval of time. Example, mangoes, fruit, all the fruits and the vegetables, it include in the organ senescence. Shoot senescence, whole aerial part of the senescence at the end of growing season, and nutrition are transported to underground storage part for survival. Example, Eutrica rumex. B is the flower senescence. It occurs after fertilization or after fruit ripening. C is the leaf senescence. Leaf senescence occurs in two ways. First one is the simultaneously or synchronous senescence. Second is the sequential senescence. Simultaneous senescence, leaf shed at particular time of growth. Example है deciduous trees. जैसे कि हम कहते हैं पतझड़ में मोस्टली जो है आपके पत्ते गिरते तो पर्टिकुलर टाइम पीरियड में होगा. दैट इज द साइमल्टेनियस सेनेसेंस. सेकंड इज sequential senescence. Sequential senescence, it is progressive senescence of the plant. It occurs, example, old leaves, branches throughout the year they are the shed from the plants. It favors development of new leaves and bud at the cost of the senescence. Fourth is the whole plant senescence. This senescence is associated with the production of fruit, which induce senescence in leaves, stem, and finally in the whole plant. Example of monocarpic species. Annual cereals, mustard, shrimp crops आते हैं. Second is biennial plants, cauliflower, radish, perennial plant, bamboos. These are the example of whole plant senescence. Then senescence जो है चार टाइप की रिकॉग्नाइज की गई जो हम आते हैं. ओवरऑल सेनेसेंस, टॉप सेनेसेंस, डेसिडुअस सेनेसेंस एंड प्रोग्रेसिव सेनेसेंस. In overall senescence, this process occurs annually, where whole of the plant is affected and dies. Second is top senescence. In top senescence, only the above ground part and root system and underground shoot system remaining the viable. Example, perennial herb. Third is deciduous senescence. In deciduous senescence occur in all leaves simultaneously, but bulk of stem and root system remain alive. Example, wood deciduous plant. Fourth is the progressive senescence. It is characterized by gradual process of senescence and death of leaves from the base upward as plant growth. Example, monocarpic senescence. जैसे कि डायग्राम है, ओवरऑल सेनेसेंस में ये क्या कह रहे हैं कि जितने भी हम देख रहे हैं जो ब्लैक नहीं है, यानी कि पूरा का पूरा प्लांट जो है डेड हो जाता है. जबकि टॉप सेनेसेंस में जो रूट और अंडरग्राउंड वाला पोर्शन है वो आपका लाइव रहता है, बाकी जो है डेड हो जाता है. जबकि डेसिडुअस में आपका रूट सिस्टम और आपका स्टेम सिस्टम ये अलाइव रहता है और इसकी लीव्स जो होती है वो डेड हो जाती है. फोर्थ आपका आता है प्रोग्रेसिव सेनेसेंस। प्रोग्रेसिव सेनेसेंस में रूट भी आपकी स्टेम भी और लीव्स भी अलाइव रहती है, लेकिन पर्टिकुलर लीव्स जो होती है गिर जाती है। तो दैट इज मतलब टाइम के अकॉर्डिंग, जैसे हम कहते हैं कि कभी ब्रांच गिर गई है, पत्ते गिर गए हैं, तो दैट इज कॉल्ड द प्रोग्रेसिव सेनेसेंस। ये वाली प्रोसेस जो है पूरे साल होती रहती है। नेक्स्ट आपका कैरेक्टराइजेशन ड्यूरिंग सेनेसेंस। तो फर्स्ट वन इज द मेटाबॉलिकली वेरी एक्टिव होते हैं ड्यूरिंग द सेनेसेंस। सेकेंड, ड्यूरिंग सेनेसेंस रेस्पिरेशन रेट जो है इंक्रीज हो जाता है, फोटोसिंथेटिक रेट डिक्लाइन हो जाता है, एंड ऑर्डरली डिसइंटीग्रेशन ऑफ द मैक्रोमोलिक्यूल्स। थर्ड चेंज ड्यूरिंग सेनेसेंस इज द क्लोरोप्लास्ट आर द फर्स्ट organelles to be disintegrated. Fourth is during senescence down regulated genes डिक्रीज हो जाते हैं। ठीक, एग्जाम्पल साइटोकाइनिन डिक्लाइन। तो साइटोकाइनिन क्योंकि सेल डिवीजन में हेल्प करता है, तो अगर ये डिक्लाइन हो जाता है तो सेल डिवीजन जो होते हैं कम हो जाते हैं और न्यू आपके जो टिश्यू है वो फॉर्मेशन नहीं होती। फोर्थ, expression of senescence associated gene increase, which encoded heterolytic enzymes such as protease, ribonuclease, lipase, जो कि ब्रेक डाउन करता है प्रोटीन्स का आरएनए डीएनए का, और लाइपेज आपका करता है लिपिड का। तो इनका ये जो है ब्रेक डाउन हो जाता है। ये जितने भी ब्रेक डाउन होते हैं ये हार्मोन सिंथेसिस में हेल्प करते हैं, सच एज द एबीए और इथाइलिन। तो एबीए और इथाइलिन जो होता है वो आपका सेनेसेंस इंड्यूसिंग करने में हेल्प कर देता है। देन 6 नंबर आता है सेनेसेंस एसोसिएटेड जीन इंकोडेड एंजाइम दैट आर इन्वॉल्व इन कन्वर्जन एंड demobilization of nutrients and substrates from senescing tissue and their reallocation to another part of the plant that survive. It means that कि आपकी जो ब्रेक डाउन हो रही हैं जितने भी सबस्टेट और न्यूट्रिशन है वो रीमोबिलाइज होंगे। जो टिश्यू आपका सेनेसेंस कंडीशन में जा रहा है वहां से जो आपका न्यूट्रिशन और सबस्टेट है वो कहीं दूसरे पार्ट में चला जाता है, जिसको प्लांट उसको फिर से रीयूज कर लेते हैं। Then 7th point is the brilliant color are developed in leaves of many plants during senescence. 8th point is more stable carotenoids pigments प्रोड्यूस होते हैं। आपको पता है कि मोस्टली जो लीव्स होते हैं वो आपकी ग्रीन होती हैं, तो जब सेनेसेंस होता है तो उनका कलर जो होता है चेंज हो जाता है, तो ड्यू टू मोर स्टेबल कैरोटिनॉइड पिगमेंट्स। 9th, टुवर्ड्स द एंड ऑफ सेनेसेंस सेल्स एंड टिश्यू जो होते हैं also lose respiratory controls. Then mechanism of senescence. So there are 4 mechanisms of senescence. First is the nutritional theory, second is hormonal theory, third is suicidal theory, fourth is the environmental theory. First one is the nutrition theory. This is due to the deficiency of the nutrition. Fruit and growth tip has strong link for translocation than leaves, so store more nutrition. Older leaves face starvation, so they fall down. Second is hormonal theory. Hormonal theory produced in developing seeds and translocated to vegetable parts to cause senescence. Presence of ABA and ethylene, both hormone induce the senescence. Third, deficiency of oxygen and cytokinin may also induce the senescence because both these hormones help in the development of the plants. Third is the suicidal theory. In suicidal theory, senescence leads progressive loss of membrane bilayer, which result in loss of permeability and leakage of the solute. Second, breakdown of tonoblast, which release the hydrolytic enzyme which digest the cellular materials. Environmental factors, stresses. Third, environmental factors such as stress. Water stress है, this create two ways of senescence. First one, accumulation of ABA, which produce senescence and ultimately cause the senescence. Second way of the water stress is reduce transport of cytokinin from root to leaves. It reduces anti-senescence hormone and senescence occur in the plants. Factor affecting senescence. So, external factor and mental factor. Second is internal factor or autonomous factor. The external factor include stress, temperature, drought, ozone, nutrition deficiency, wound, darkness, salicylic acid and the shedding. Internal factor includes age, reproductive development, level of phytochrome and sugar, decline in the photosynthesis. So these endogenous and exogenous both factors cause the leaf senescence. During leaf senescence, there are different phases such as the shifting in gene expression, chlorophyll degradation in protein, lipid and other macromolecules, nutrition translocation by the phloem, cell death is the ultimate phase of the senescence. Second, drought, stress, it increases the ABA concentration. ABA concentration causes the three way of the senescence. First one is the regulation of ion channel activity, which close the stomata and increase drought or desiccation tolerance. Second, regulate of ABI transcription factor activity, cause the dormancy in the seeds and increase the drought rates. Third, increase ABIG1 transcription, which regulate the target mRNA. It inhibits the leaf senescence and cause the drought-induced senescence. Then physiological and biochemical change during senescence. So, first one is the disrupt cell functioning. Second is reduced photosynthesis. Third, breakdown of chlorophyll. Fourth, loss of chlorophyll pigments and shows the yellow leaf. Fifth, change in metabolism of protein and lipids. Sixth, lipid converted into soluble sugar by gluconeogenesis pathway. It changes the membrane permeability. Seventh, decrease RNA content and ribosomal degradations. Eighth, nucleic acid breakdown and chromatin material properties are also changed. Ninth, increased respiratory rate, but in the terminal phase of senescence, this respiratory rate is decreases. Physiological and biochemical change. The first one is increasing respiratory rate. To increase respiratory rate because continuous supply of energy is needed for sustaining degradation of cell structure. Second, mobilization of nutrients from the senescence organ to the another part of the plant. Second, ethylene is responsible for enhancing plant respiration by activating pre-existing enzymatic potentials. Note, respiratory decrease in senescence leaves, which increase catabolism and decrease anabolism. Activity of hydrolytic enzyme increase, but synthetase is decreased. Second, chloroplast membrane degradation. Rubicoscope present in the leaves, that is degraded. Due to this, increase in free radical productions and ultimately is destroy the membrane. Second, bulbing is stopping. In senescence, selective exchange by the membrane is losses. Due to this, particle accumulation between lipid biliaries occur and it causes the phase separation and ions leakage and ultimately membrane further degraded. Second, CO2 assimilation decrease. Low CO2 cause closing of stomata. Second, decrease photosynthesis. If it closes the stomata, then transpiration decreases, or if photosynthetic rate is decreased, then less photosynthetic assimilations is occur. Photosynthetic pigment. Chloroplast. Photosynthetic pigments. Breakdown of chloroplast. It occurs in order: chlorophyll A, then chlorophyll B, and then carotenoids. Degradation occurs by the different enzymes such as the chlorophyllase, chlorophyll B reductase, and chlorophyll oxidase. Methyl jasmonate also induces the chlorophyll degradation. So, chlorophyll A, in presence of chlorophyll oxidase, chloroplast degradation occurs. Methyl jasmonate strongly favors the increase in the chloroplast degradation. Methyl jasmonate, it induces lipid degradation, also induces the senescence expressed related of genes such as the cysteine and protein genes. Internal factors such as the phytohormones, including cytokinin, auxin, ethylene, jasmonic acids, ABA, salicylic acid, stickolactones, and reproduction. Second, external factors such as ultraviolet B, ozone, nutritional limitation, heat or cold, drought, shading, pathogen attacks, and wound attacks. Also, another factor that is the developmental age. They induce the signal transduction, and transcription factors such as SNAC, VARK, and others. They are induced and send the signal to the plant. During this one, senescence inducing genes, they are increasing concentration and degrade the nutrition cycles and leaves become the yellow. As well as senescence degraded genes, they increase the photosynthetic biosynthesis and green leaves and leaves जो हैं help करते हैं development. Nucleic acid, 20% DNA is degraded in late phase of senescence. Example: tea, tobacco, soybean. Intracellular proteins, increased breakdown of protein caused by the de novo synthesis of proteolytic enzymes as well as the activation of pre-existing enzymes. Fourth, free radical formation and antioxidant enzymes. Example: alkyloid radicals, peroxides, superoxides, hydroxyl, ROI, reactive oxygen intermediate. Reactive oxygen intermediate, it causes the oxidative damage in the late stage of the senescence. They are produced due to impairment of electron flow between two photosystems, which limit the availability of the photosynthetic power. Free radicals, they cause the breakdown of proteins, polysaccharides, nucleic acid. Second, enhance ethylene production pathway. Third, initiate chain reaction of membrane, which led to extensive peroxidations. As a result, alternation of fluidity and permeability of membrane take place. Fourth, decreased plasma membrane permeability, loss of membrane integrity, decrease in total phospholipid and protein content. Third, increase in neutral lipids and channelization of oxygen. These three steps cause the decrease in plasma membrane permeability. Fourth, enzyme involved in the lipid degradation such as the phospholipid D, phosphatidase, phosphatase, lipolytic acyl hydrolase. Delta 9 desaturase. It increases the senescence by degradation of saturated fatty acid of the membrane lipids. Sometimes this multiple questions is asking which enzyme help in the degradation of saturated fatty acid during the senescence. That is the delta 9 desaturase. Decrease plasma membrane permeability. तो इसमें डिफरेंट चेंजेस है. लिपिड फ्लूइडिटी चेंज, फेज ट्रांजिशन, टेम्परेचर चेंज, नॉन bilayer lipid structure. They all cause the bilayer instability. It leads to failure of membrane associated enzyme and receptors, which produce the unsaturated fatty acids. It causes the increased phospholipase and phosphatidic acid phosphatase. Second is change in lipid composition of membrane. It also causes leakage of calcium to positive, which lead the increased phospholipase and phosphatidic acid phosphatase, which increase the unsaturated fatty acids and ultimately oxidative injury to the membrane by the superoxide radicals and cause the oxidative senescence. Regulation of senescence by the plant hormone. There are two types of hormone regulation. First one is the senescence promoting hormones, which include the APA, jasmonic acid, brassinosteroid and salicylic acid. Second is the senescence inhibiting hormone, such as the auxin, cytokinin and gibberellic acids. Role of cytokinin in senescence regulation. Cytokinin involved in senescence, gytin ribosides, dihydroxygytin ribosides. They delay the leaf senescence. Proof: low concentration of cytokinin in senescence tissue. Exogenous cytokinin delay the senescence. Evidence क्या है? Cytokinin exert role in mobilizing nutrition. High cytokinin have strongest metabolic sink, so nutrition move. जेनेटिकली इसका प्रूफ क्या है कि साइटोकाइनिन सेनेसेंस रेगुलेट करता है कि काइमेरिक जीन्स such as the isopentyl transferase IPT gene promote senescence. Iptic genes block expression of cytokinin level. Cytokinin part plus में senescence part plus cytokinin. It delay the senescence by blockage expression of chimeric genes. Role of ethylene and ABA in senescence regulation. Aminoethoxyvinyl glycerin. It inhibits the ACC synthase, which help in the production of the ethylene. Ethylene cause the chlorophyll loss and cause the senescence. Second is ABA. ABA stimulate ethylene formations and increase the abscission, which cause the senescence. Second, ABA also increase the sugar level change and cause the senescence. ABA effect on senescence. There are different ways. First one, ABA cause calcium 2 positive open channels which depolarize the plasma membrane and promote the chlorine negative channel. It can enhance the depolarization membrane. Second, ABA induce the IP3 level increase. It releases calcium 2 positive from internal storage, which cause the increased cytosolic calcium 2 positive, which activate the opening of chlorine negative channels and inhibit inward movement of the K positive channel, which depolarize the membrane and long-term depolarization of chlorine negative efflux occur, which cause the stomata closing and stomata closing decrease the photoassimilations. Third way of the ABA effect to senescence is that ABA stimulate ethylene formation, which increase the abscission and cause the senescence. Effect of ethylene can be neglected by the high concentration of CO2. Oxygen and ethylene interact in many ways. Since high oxygen level trigger the ethylene production, while high ethylene level cause the induction of enzymes peroxidase that inactivate indole acetic acids. Molecular level of senescence. Senescence has two types of genes. First is the senescence downloaded genes. It includes all the photosynthetic enzymes. Second is the senescence associated genes such as the protease, lipase and nuclease. Senescence associated genes, they are expressed in low level. These genes whose express is upregulated with onset of the senescence, encoded for enzyme which involve in degradation of lipids, proteins and amino acids. Then protein degraded, encoded protease for the enhancing senescence. Example, Rhabdopsis thaliana, SAGE2, SAGE12, SAGE15. Mice have C1. For RNA degradations, RNA2 encoded for sRNase, expressed in phosphate starvation, so mobilization of compound during senescence. Stress-related gene, superoxide radicals, superoxide muteज की प्रेजेंस में H2O2 की फॉर्मेशन करता है, which cause detoxification, and produce catalase, peroxidase, and enzyme of ascorbase glutathione cycles. So overall, this one is that internal factor and external factor. They both cause the developmental age, which cause the onset of the senescence. During senescence, macromolecules degraded, nutrient salvation and translocation occur. And third is the detoxification and defense. They all cause the chlorophyll loss, nitrogen lipid mobilization, increase of antioxidant and defense related genes, which cause the cell death. During cell death, DNA leading or disruption of nucleus and mitochondria occur. Second is disintegration of plasma membrane and vascular membrane occur. Significance of senescence. First is the self-maintaining process. Second, reduced transpiration rate and export metabolic bags. Example, deciduous plant. Third, protect plant from wastage of nutrition by send it to underground part. Example, biennial or perennial plant. Fourth, save expenditures of nutrient in old organs. Fifth, for proper functioning of tissues such as xylem elements. Fifth, live senescence. Adaptive value to complete their life cycle under the stress condition. So these are the different significance of the senescence.